My name is Mirko Rossi and working at the European Food Safety Authority in Parma. Today I'm going to present you the data from our poster here in IMMEM. The European Food Safety Authority and the European Centre for Disease Control and Prevention have implemented since 2022 as a response from European Commission mandate the One Health WGS system. The system has been designed for collecting CG/MLST profile from human and non-human environment and to exchange this profile seamlessly, programmatically, and to develop specific dashboards and tree and other type of representation to be then given to the Public Health and Food Safety Authority member states and to investigate, to support the investigation of foodborne outbreak at EU level. The data arriving to our system is on voluntary basis, and we are collecting data on Listeria monocytogenes, Salmonella enterica, and E. coli. The voluntary submission is also coupled with a mandatory submission from the food safety sector. The mandatory submission have been laid down to the new regulation that have been going to be enforced starting from next year in August 2026. If you want more information on the EU regulation, you can use this kind of link and where you can find the FAQ from Commission. The main important point for the new regulation is that the member states are now mandated to submit data to EFSA in case of food isolate of species of Salmonella enterica, Listeria, E. coli, Campylobacter, Campylobacter coli, when these have been found suspicious or associated with foodborne outbreak. Nevertheless, we were able to collect a lot of data in the recent 3 years in both sectors, reaching approximately 37,000 data, even more recently. This is grow up on a weekly basis. This kind of data was able to identify over 300 genomic clusters across different sectors, and was an important finding for supporting a EU agency and member state to analyzing and assessing over 10 major EU outbreaks, mainly for Salmonella and Listeria. What I would like also to underline is that we are using CG/MLST as a main methodology for performing genomic surveillance. The capacity of the two systems to share seamless data is due to the fact that we are not using a centralized nomenclature server, but we translate the Lely profile in hashes, and this has helped us to just seamlessly integrate each other and exchange only what is needed for the actual definition of the cluster and the identification of a common food possible source. This initiative is, in our opinion, one of the most best example for One Health integration within the European Union. We are very important for us to also underline that one of the critical points on this implementation is the capacity to maintain the chain of custody of the data that we are receiving. This allows us to have a secure data use and storage, and also allowing the member state to remain full ownership on the data within our system. I would like to thank our colleague at the European Food Safety Authority and the European Centre for Disease and Control and Prevention, and also all the member states for participating in providing the data.